My name is Alan, and I will be your host for today's webinar entitled The Complete Transgene Integration Site Characterization for Producer Cell Banks and Immunotherapy Products with TLA Technology. So as promised, I'll be touching upon the relevance of our TLA analysis within the cell and gene therapy space and how our TLA has essentially been able to help and support many of our customers in their work within this field. But first things first, a little bit of background information about our firm. So we're a Dutch genomics company, and we were founded in 2012. We're actually a spin-off from the Hubrecht Institute, which belongs to the Royal Netherlands Academy of Arts and Sciences. And the Hubrecht is a biomedical research institute that focuses on developmental biology and stem cell research. So we are based in the heart of the Netherlands in the charming historic city of Utrecht, which is only about 20 minutes away by train from the capital Amsterdam. And more specifically, our office is nested in the Utrecht Science Park itself, as you can see here on the map. But what do we actually do? Well, we've essentially pioneered this TLA technology that we offer both as service but also kits. TLA, as you have guessed, is an acronym. It is short for Targeted Locus Amplification, and that's essentially what it does. So it allows the complete targeted sequencing of any gene or transgene of interest. So to date, there are, of course, many different techniques currently being used to generate transgenic cell lines. However, all these techniques can result in undesired off-target integrations, multiple integration sites, unexpected integrations of backbone sequences, or even in undesired sequences or structural variants in the integrated transgene itself. So generally, people that come to us place a lot of importance on the thorough characterization of their genetic engineering. So as you can see here on this slide, we have listed here all the different criterias and characteristics that many researchers typically take into account and find important to evaluate when creating transgenic cell lines or when simply editing specific loci. Now, of course, if the intention is to sequence the entire genome for mutations that might be linked to the trait that you're interested in, then WGS is the way to go. However, once you actually know the genes that are relevant to you, or when you simply know the transgene sequences that you're working with, then whole genome sequencing approach can be a rather inefficient way of generating sequence information across those regions of interest. Because indeed, you would here be generating such large volume of data, most of which will essentially be quote unquote junk, thereby rendering this approach rather superfluous. And on top of that, generating such large volume of data will render the analysis and interpretation of the data even more complex. So this approach in this particular scenario would not be very time efficient nor cost effective. And also because you're generating sequence information across the entire genome, this will result in lower coverage, meaning that a fewer reads will span the region of interest, thereby affecting sensitivity. So the sensitivity with which you can detect rare mutations, rare sequence changes is actually going to be lower. Now, in terms of PCR, of course, you're amplifying here regions in between the primer pair that you're using. It's, of course, a great tool and an effective way to pick up, for instance, point mutations or small indels. However, if the genes of interest is actually large, then this approach can become rather cumbersome, especially if unexpected mutations have occurred there. Indeed, with PCR, you will inherently miss anything that you're unaware of, given the hypothesis-driven nature of the technology. And finally, southern blot and FISH analysis are, of course, often used for the genetic analysis of generated cell lines and to get an idea, for instance, on rearrangements or integration sites, respectively. But the data generated here are incomplete. So I hope that this table makes it somewhat apparent as to how none of the conventional technologies listed here can, in a single experiment, yield reliable information on all of those different characteristics. So the main takeaway message of this slide, I would say, is that despite the advancements in targeted gene sequencing and whole genome analysis, that the robust detection of all genetic variations, and this including structural variants in and around genes of interest, still remains a challenge. And this is how our TLA actually sets itself apart and represents a truly unique alternative. So how does our TLA actually work? Well, the simplest way I like to explain this is using a single primer pair, we will be able to uncover any gene or transgene that you're interested in. But we can also with TLA sequence neighboring regions, and these regions will be of sizes ranging from tens to hundreds of kb. So essentially, we'll be able to generate comprehensive coverage over large regions spanning from the position of those primers. So how do we go about this? Well, when we receive the samples from our customers, we start by cross-linking them. For this, we use formaldehyde as a cross-linking agent, and that's what we try to depict here using this blue dot. We then proceed to digest the DNA using restriction enzyme and then religate to create circles. So essentially, we reshuffle the DNA to create circles that then contain fragments of the digested DNA. But what's interesting about our TLA technology is that it actually uses the principle of physical proximity as a basis of selection, meaning that sequences that were originally close from one another will have a higher chance of ending up in the same circle. Still, it's still very much a stochastic process. So all that we've done until now is simply convert the genome into these circles, or TLA template as we call it. And what comes next, obviously, is the enrichment step. So there, we will actually design primer pairs, or should I say inverse primer pairs, that will be actually complementary to a short specific sequence of your locus of interest, or your transgene. And by doing this, we selectively amplify the circles that we're interested in such a way that in the end, we'll get something like this type of high sequencing coverage profile. So it looks, as you can see, somewhat like a bell-shaped curve. And again, the coverage will be highest in the immediate vicinity of the prime pairs that were used, and the coverage will decrease and decline with greater physical distance from the position of those primers. So because of this, TLA can actually be relevant in a number of different things. And the different points listed here are typically the things that our customer come to us for. So to help them determine the number of integration sites, determine the genomic position of those integration events, but also to help them assess where the structural changes, so be it point mutations, SNVs, or larger structural variations may have occurred in and or around genes of interest. So this is our landmark Nature Biotech paper where we have published for the very first time the proof of principle of our TLA technology back in 2014. So in case you're interested in, I'd recommend you to visit our website and you'll be able to get a direct link to this paper. Now, in terms of integration site, as I've mentioned, we will not only be able to detect single, but also multiple integration sites as shown here. So to give a little bit of background information as to what you're looking, these are what we call whole genome wide coverage plots. Essentially, on the y-axis, we stack on top of one another all the different chromosomes, as you can see here. On the x-axis, you'll find the chromosomal position, and basically, following TLA preparation, amplification, sequencing, and then mapping all the reads back onto the reference, we get this peak and coverage at what appears to be here, chromosome 8, therefore suggesting a single integration event taking place. However, as I've mentioned, we will also be able to detect multiple integration sites, as shown here on the right-hand side of the slide. So as you can see, there appears to be a total of 3 integration events in this particular case at 3 different positions on 3 different chromosomes. Now, in fact, we will be able to identify integration sites at base pair resolutions on a single nucleotide level, but we will then also be able to detect the breakpoint, so the breakpoint between the transgene and the flanking host genome. And because of that, we'll also be able to help you and or your colleagues assess whether larger structural variations may have accompanied your genetic engineering. So in case you're confronted with a deletion, duplication, inversion, or translocation, these are the type of aberrant coverage profile that we would expect to pick up in our analysis. So just to elaborate a little bit further on what you're looking at, so in case you're dealing with a deletion, then we would expect to see a gap in the generated coverage following integration of the transgene. In case of duplication, we would then expect to see the sequence being duplicated at either end of the integrated transgene. In the event of inversion, we would expect to see this type of aberrant coverage profiles whereby the arrow actually depict the position of the breakpoints. And for translocation, of course, you have on one chromosome the transgene, then the breakpoints, and which then continues onto a different chromosome. Now, to demonstrate the capabilities of our TLA, in this case, TLA was used to sequence a gene that's about, as you can see, 10 kb in size. As you'll notice also, the coverage here is 1000x. And I actually would like to draw your attention to the middle part of the figure. So this is actually a limitation of NGS. So as you've probably understood by now, TLA is actually used in combination with next generation sequencing, and NGS, as you know, struggles whenever it runs into a region that's rich in AT or CG repeats, thereby leading to a drop in coverage. So basically, that's what you're seeing here. However, as you can see, it's still rather a sensitive approach to sequence entire genes. Again, in this case, it was about a 10 kb size gene to pick up also all mutations within. Of course, I'll support this claim with the coming slides. So we will, with TLA, be able to reliably detect SNVs. So in this case, there appears to be 2-point mutation with regard to the reference, but we will also be able to detect infrequent SNVs as a point mutation in a subset of the reads. So in this case, it appears that about 20% of the reads or so have this point A mutation with respect to the reference at this particular position within the transgene. It is actually good to note that as part of our standard analysis, we typically use 2 primer pairs, and there are 2 reasons for this. So if we suppose that this transgene, and I hope that you can see my mouse here, if this transgene were to partially integrate on this end, then what we call here primer pair 1 will be able to pick it up and not primary pair 2, but also vice versa. And the second reason is just that it's a good way for us to QC any genetic variants within the transgene in such a way that we will be able to share with you and your colleagues these type of tables. So here we show the mutation, the position at which it occurs. We list the reference. We put side by side the data generated by each of the primer set, showing the frequency at which the mutation has occurred, as well as the associated coverage. Now, of course, as you can imagine, in the absence of large structural variations, we would then expect to see these numbers to be more or less in line with one another. And finally, in terms of concatenators, well, we will be able to identify those. So in the event of partial or multiple copies of a transgene co-integrating together, we will be able to pick this up. In fact, we will report this as transgene and transgene fusion. And as you can understand, these has important consequences in terms of the expression of aberrant proteins. So now moving on into the relevance of our TLA analysis in the upstream process of cell and gene therapy. Well, TLA can actually be applied at different stages. Well, first of all, in the optimization and validation of new technologies. So for instance, you might be interested in identifying the hotspots of targeted integration, but even if you're working with random integrations, you'll probably still want to know where the integration has ended up in the genome and if the integration is clean and so on and so forth. So TLA will be able to help you assess the genetic consequences of the different techniques that you're working with to really help you further fine-tune the appropriate tools to be used in your final actual production process. Secondly, TLA can also play a key role in clone selection. So this is thanks largely to our ability to output all of those different characteristics that I've already mentioned early on. So in terms of identifying integration sites, SNVs, or larger structural variants in and around genes of interest, but as well as picking up potential concatenators and giveaway copy number estimation. An illustration of our work here is this paper that was released by Novartis last year, describing the relevance of our TLA as part of their CHO clone screening towards their manufacturing of their monoclonal antibody. Now, in terms of our monoclonality assurance as well as genetic stability assessment, we actually have the pleasure to support many of our customers in these type of endeavors. So here are just 2 concrete examples together with Glenmark and Gilead, as you can see. And here, basically, using TLA, we were essentially able to conclude that Glenmark was working with clonal and stable CHO clones. And similarly, we investigated about 30 research cell bank subclones and concluded that Gilead was also working with highly genetically stable clones. And with that said, I actually would like to highlight that Glenmark has now filed a patent claim on the statistical approach described in this paper that you see here. Nevertheless, we have recently released a white paper together with Novartis using an alternative statistical approach, and again describing here the relevance of our TLA towards clonality assessment. And while similar for clonality, TLA assesses genetic stability by first performing TLA on the MCB in order to determine the genetic makeup of this line. And subsequently, if TLA analysis of downstream clones also yield the exact same integration site, the exact same concatenators, and the exact same mutational profiles, then we can conclude that the cell line is genetically stable. So as you'll notice, I have until now only discussed about papers that are mainly tied to our CHO work. However, as you can understand, there are a lot of overlap and similarities with the cell line development process for the production of gene therapy products. And in fact, this now brings me to my next slide, and we are extremely excited to be sharing with you today these two new publications by GSK, as well as a collaborative work between the FDA and NIH researchers. So as you well know, lentiviral vectors are actually typically produced using the human embryonic kidney 293 cell line. However, the presence of SV40T antigen encoding sequences actually raises some safety concerns regarding the use of those cell line to produce viral vectors for in vivo applications. So what the researchers from the FDA and NIH have done here is that they've removed this SV40T antigen encoding sequences from the HEC293 cells using CRISPR strategy. And TLA was actually one of the selected methods to help perform in-depth characterization of this genetic modification. And another very interesting paper by GSK here also, where they essentially reported a method to rapidly generate producer cell lines from 293 cells via the stable transfection of a single DNA construct that encodes all lentiviral vector components. So this is again another very nice illustration of our experience with analyzing a variety of different cell lines, because in this particular study, TLA was actually used as the method of choice to map integration sites of their back constructs. Now, another very elegant paper that was published by a team of researchers from the University Medical Center of Utrecht, right next door to us. So they've actually characterized here not only their downstream CAR-T sample with TLA, but also their upstream producing cell lines. So the rationale for this actually came earlier on because the researchers involved in this project actually realized that there was, at least to the best of their knowledge, no comprehensive information available regarding the molecular characterization of integrity of CAR or TCR transgenes on the nucleotide level throughout the complete production chain as requested by regulatory authorities like the FDA or EMAs. So they realized the scarcity regarding the availability of those important information in terms of thorough genetic characterization, as I've said, from vector producer cells until final medicinal product. So we were here able to reliably detect several SNVs with TLA, but for the sake of time and for simplicity, I've only highlighted two. So we found in their downstream eventual product two mutations, in fact, two single nucleotide substitutions, as you can see here. But what they also did, as previously mentioned, is that they also submitted their MCB, so their vector producing cell line, as well as their plasmid for TLA analysis. So what we were able to do with that is that we were able to pinpoint where those SNVs were likely to occur. So looking at both the final downstream product and the vector producing cell line, MCB, we actually observed that those mutations were actually popping up at about the same ratio, so between 10 to 15%. But interestingly enough, those variants were actually not detected in the plasmid, suggesting that these variants actually likely arose from the integration of the vector producing transgene into the MCB. And as previously explained, we can also with TLA determine the integration sites. So here we found a total of 9 integrations in those cell lines of the MCB viral producer cell lines, indicating that likely one of the copies in here contained the SNV. So in terms of our relevance in the downstream application, this is a very nice example together with one of the groups at the Memorial Sloan Kettering Cancer Center. Well, in this study, they actually wanted to target a CAR to the track locus using CRISPR-Cas9 strategy. So using TLA in the same manner that I've depicted until now, basically with the primers complementary to the transgene, we were able to confirm a correct intended targeting. So again, you're looking at a whole genome wide coverage plot, and it appears to have a single integration event at this particular position on chromosome 14, suggesting then correct intended targeting. But what we also did in this project is that we placed the primers right next to the transgene. So this is very interesting because it allowed us to generate sequence information also on the wild type allele in such a way that we were able to actually quantify the number of correct but also incorrect integrations at the intended locus. So at the end of the study, we were actually able to conclude that 80% of the alleles actually were wild type, meaning that they did not undergo rearrangement, modification. but 20% did. And of those 20%, 75% actually had perfect integration, homologous recombination, while 25% did not. And so in this matter, we were able to demonstrate in the study and through this work, the suitability of TLA to assess the efficiency as well as the accuracy of targeted integrations. Now, this is another recent paper together with Selectus. So here, researchers at Selectus were actually looking at T-cell in the studies, whereby they actually had used a dual targeting approach, so TALENs and HTR, to integrate two different transgene in a single sample to either chromosome 14 or 2. So the hypothesis, of course, was that integration would only happen at either of those target sites using homology arms. And so we use TLA primers on either of those vector sequences. So when placing primers on the transgene that was supposedly integrating only on chromosome 14, we actually then observe a single peak, single coverage peak there as intended. So that was good news. And this was actually also cross-validated with other approaches like FISH. However, we also actually saw another peak on chromosome 2 where the other target site was. So this actually indicated to us that some rearrangement had occurred between these two sites. Now, and of course, looking at the exact single nucleotide and breakpoint sequences, we were able to exactly resolve which rearrangements these were. And when looking at the transgene that was supposedly only integrating on chromosome 2, we again found some translocation occurring between chromosome 2 and chromosome 14. And in conclusion, as you can see, with TLA and with only one or two primer pairs, we can, in a single experiment, resolve the exact genetic characterization of your edited locus or integrated transgene sequence. Now, with regards to the analysis of random integrations in heterogeneous samples, we will actually not identify the integration site based on those whole genome wide coverage plot that I've shown you earlier, but we will detect those integrations based on the detection of the breakpoint sequences. So just to illustrate this with one final example, so in this case, TLA was used to completely sequence the HIV genome from a patient's blood sample to determine where in the genome the integrations have happened. So what you're looking here on the slide are actually vertical bars. So these are just graphical depictions. These are not peaks in coverage as shown earlier, and they're just supposed to depict the position of the breakpoint. So in the study, with TLA, I have identified here around 1,400 to 1,500 breakpoints. And we do very similar work in the analysis of CAR T cells and other genetically engineered cell lines, where based on the breakpoint sequence, we are actually able to detect the positions of the integrations. So finally, I hope that I was able to convince you of the unique capabilities of our TLA technology and that this session was helpful in better understanding the power of TLA to not only efficiently map the integration site of any transgene, but to also provide additional information regarding the transgene integration events, for instance, in terms of identifying structural changes occurring at the site of transgene integration. So I've condensed here one last time all the different genetic characteristics that we're able to output with our TLA assay, just to highlight the fact that our TLA will be able to help you and your colleagues fully characterize integrations of your transgene or vector, as well as obtain all essential information on the integration sites, mutations, but also potential concatenators unlike conventional technologies. And with that said, we have almost a decade of experience analyzing a variety of different cell lines and have the pleasure to support customers ranging from both academia, but also leading biotech and pharmaceutical companies globally. So in case you might be interested in learning more about our TLA technology and our company, I would kindly invite you to check out our website, and please feel free to reach out to us anytime by email should you have any additional questions. We're always happy to help or schedule a call together with your colleagues or your team to learn more about your work, but also to discuss it a bit in more detail if and or how our TLA analysis can best support your work. So with that, I would like to conclude and thank you all for your attention today.